How's everybody doing?
Good.
Next to last day of class. Only 2 times 50 minutes more opportunity to cram information into your heads. So a couple of announcements. So number 1, if you've turned in an exam for regrade, I will have it back for you on Friday. You can come pick it up from me. Number 2, I announced last time that the exam would not be comprehensive. It would, in fact, be a just material since the last exam. No note card as a consequence. And that's only for this classroom class. The eCampus class will have a different set of rules when they see this video in the future. One student asked me beforehand, and I haven't decided on this yet, will it be a 50-minute exam or what will it be? I'm inclined to make it not a 2-hour exam. That's my gut feeling, because my experience with this is that if I asked you to write your name, there would be people who would spend 3 hours if I would let them. So I think a reasonable amount of time for this exam will be about an hour and 20 minutes. Okay? And it won't be any longer than the other exams you've already seen. Okay? So and time, I don't think, has been a factor for any of the exams. So I'm tentatively planning, I'll announce it on Friday, but tentatively planning for an hour and 20-minute exam. Okay? That should give you plenty of time to look over things and make sure you've got what you need. Okay? Let's see. And review session. That was the last thing. Yeah. Location exam's right in here. Yeah. And that is on Monday at 2 p.m. in here. Okay? The review session, I will tentatively plan for it on Saturday. And I'm guessing late afternoon, but I haven't decided. So that will require getting the building opened and all that, and see how that goes. So I'll check on that. Okay? Comments, questions?
Will you post the video by Sunday?
Will I post the video for the review session? Yeah, I will, and I will try to get it posted that evening, yeah. Okay, so we're moving along. Nitrogen metabolism. Nitrogen metabolism is, I think, an actually underrated aspect of metabolism. A lot of people look at nitrogen metabolism and they think, oh, it's urea and it's that sort of stuff, and it's just not much interesting and that's not much fun. It's just waste metabolism. But in fact, nitrogen metabolism, for anybody interested in nutrition, which many of you are, is very critical. Because nitrogen is a necessary nutrient, and nitrogen is an essential component of amino acids. Okay? And the ways in which our bodies and in which cells handle nitrogen relative to amino acids is interesting, and it's very important. So I want to start in very general terms talking about nitrogen metabolism, and then get down to some specifics. We've all heard about the nitrogen cycle. There's a nitrogen cycle in the world that exists. And nitrogen is, in terms of nutrients, a very unusual one. Okay? We have an absolute abundance of nitrogen in our atmosphere. In fact, the most abundant molecule in our atmosphere is molecular nitrogen. And that molecular nitrogen is completely unusable by us. Completely. We can do 0 with atmospheric nitrogen. Okay? We breathe it in and we breathe it out, and that's all that ever happens to atmospheric nitrogen. Okay? So in order for us to use nitrogen effectively, it has to be converted into a form that can be handled by our cells. And here's a place where I really argue that we should be very careful in thinking about polluting our environment and damaging our environment. Okay? We being higher organisms, we are ultimately totally dependent upon microbes for nitrogen. Plants can't pull it out of the atmosphere. We can't pull it out of the atmosphere. Okay? Most bacteria can't pull it out of the atmosphere. There's a relatively small number of organisms that can use nitrogen and then convert it into a form that we can use. As we go polluting soil and water and damaging these microbes, what we're ultimately doing is damaging our ability to use nitrogen. And when we run out of nitrogen, well, there isn't going to be any life because no nitrogen, no amino acids. Okay? Well, this figure is pretty general. It shows overall what has to happen. And ultimately what we're trying to do, as far as we are concerned, is get to something that is a reduced form of nitrogen. That's really important. That's what we're trying to get to. We're starting with atmospheric nitrogen and through a variety of places getting down to usable nitrogen. The nitrogen that we have on Earth, and there's other mechanisms, but the most common mechanism of getting molecular nitrogen is a reaction catalyzed by an enzyme known as nitrogenase. And nitrogenase is an enzyme that is very, very, very exquisitely sensitive to molecular oxygen. So it requires anaerobic conditions, and it's only present in certain bacteria. And it is a very energy-intensive process to convert molecular nitrogen ultimately into ammonium, which is a reduced form of nitrogen that we can actually use. Okay? There are a total of 16 electrons that have to be managed in the process of reducing molecular nitrogen. Now, I'm not going to take you through how microbes do that, all right? But suffice it to say that it's a complicated process, and it's a very energy-intensive process, okay? There's at least a dozen ATPs necessary to get nitrogen reduced, all right? When we get it reduced, we want to be able to do something with it, and or we want an organism to do something with it. Of course, we don't have these organisms inside of us, so we're getting it through the food that we eat and so forth, and those plants and animals are getting it ultimately from these soil microbes that are assimilating atmospheric nitrogen. Well, what happens, what's one way in which I can take ammonium and use it? Well, this shows very nicely. Here's one reaction that can do that. Okay, so there's ammonium ion right there. That's just basically ammonia with a proton on it is all it is. And ammonium can be combined with glutamate to make glutamine. That's it right here. Okay, ammonium can combine with glutamate and make glutamine. Now, if you remember your amino acids, you will remember that glutamine had not only an alpha amino group, but it had a carboxyamide. That is, it had an amine group on its R-chain carboxyl. Okay? That's how it differed from glutamic acid, for example. It had an amine instead of an OH on that R-chain carboxyl. All right? Asparagine is the equivalent one. Aspartic acid, you take that hydroxyl off and you put an amine on that place and you've got a carboxyamide and you have something that has two amines. Well, glutamate and glutamine play very important roles in nitrogen swapping. A lot of what happens with nitrogen in our bodies and in the cells of any organism is basically the transfer from amine group from one place to another to another to another to where it's ultimately needed. And amino acids are carriers of this, and amino acids are necessary for us to have this nitrogen. So we see that one way we can assimilate that is with ammonium. We see that that, too, is a process that takes ATP. And we see a variety of regulation of this enzyme. Okay? There's a lot of different things that can negatively affect this enzyme. Okay? Serine, histidine, tryptophan, okay? Carbamyl phosphate, AMP, CTP, glucosamine 6-phosphate, all negative regulators of this enzyme. Okay? It tells us that it's important that cells control how much of the ammonium or how much ammonia that, or a reduced nitrogen that we're actually taking in and using. If we have too much, that's not good. Okay? So we'll see that one of the ways our body handles too much is in what's called the urea cycle, but that's coming up. This shows the progression in going from alpha-ketoglutarate, which is what this guy is over here. That was from the citric acid cycle. We put an amine on there and we make glutamic acid or glutamate. We go from glutamate to glutamine, and that's now replacing that Here's the carboxyl that was on glutamate. Here is the amine that has now replaced it on the R group on the end. Okay? This process that you see here is important in, again, nitrogen metabolism. And what we see in these sets of reactions are examples of what are called transaminations, particularly this top reaction. Now, I've mentioned transaminations before, but I haven't really told you about them, so I want to say a little bit about them. Transamination reactions are ways of swapping amines with oxygens. They're ways of swapping amines with oxygens. Okay? Well, this itself is an acquiring of an amine group. It's not on its surface a transamination, but I'm going to give you an example of something that is a transamination. That could work for here, and that could work for here as well. Let's imagine that I have made glutamate. Okay? So I've assimilated this ammonium, and now I've got glutamate. I could take glutamate to glutamine, but I might also want to, let's say, convert pyruvate into alanine. How do I convert pyruvate into alanine? Well, pyruvate has a double-bonded oxygen. And transamination means that if I can swap out that double-bonded oxygen for a nitrogen, I've just made alanine. So the only difference between pyruvate and alanine is that alanine has an amine group, pyruvate has a double-bonded oxygen. Okay? All right. Okay, oh, I didn't have what I want there. Okay. So, I need, if I'm going to make pyruvate and convert it into alanine, I need an amine group. Where's the amine group going to come from? It's going to come from right here. Okay? Now, let's think about this. Pyruvate has an oxygen. Glutamate has an amine. If I swap the amine with the oxygen, what am I going to have? I'm going to make alanine. If I put an oxygen in this place, I'm going to make alpha-ketoglutarate. Okay? So this is the example of what I'm talking about with respect to a transamination reaction. All right? I'm taking an amine group from one amino acid and swapping it with an oxygen from another amino acid. In turn, I convert the amino acid into an alpha-keto acid, alpha-ketoglutarate. When I convert alanine, I can go the other way. I can take alanine and I can have it donate its amine group to alpha-ketoglutarate and make glutamic acid. What I'm left with then is pyruvate. And pyruvate is an alpha-keto acid. These reactions all involve swaps involving an alpha-keto acid. Okay? I can do the same thing with aspartic acid. If I swap aspartic acid with pyruvate, aspartic acid has an amine group. I put the amine group on pyruvate, I've got alanine. I put the oxygen on aspartic acid, and I've got oxaloacetate. Okay? That's an alpha-keto acid. Now, these transamination reactions we will see pop up in all kinds of amino acid metabolism. When we go to make amino acids, this is one of the primary ways in which amine groups make it onto alpha-keto acids to become amino acids. Very important point. So transaminations are very critical for the synthesis and swapping of nitrogens to make amino acids. Okay. All right, so you've already seen a little bit of this. When we talk about amino acid metabolism, which is what I'm getting ready to talk about right now, we see that amino acid metabolism is grouped into certain families. Okay? The families that we see, here's alpha-ketoglutarate going to glutamate. You've already seen that. That's an alpha-keto acid that's going to an amino acid. A transamination would give this. Okay? We could also do it by adding that ammonium group that I just showed you. Either way, we get glutamate. Glutamate can be converted by various reactions into any of these three amino acids. So glutamate is a precursor of glutamine. It's a precursor of proline. It's also a precursor of arginine. Okay? If I can't synthesize glutamate, all right, if I cannot synthesize glutamate, it means I must have it in my diet. We can synthesize glutamate. But if I can't, any amino acid I can't synthesize, I have to have in my diet. It's known as an essential amino acid. You've heard that term before? An essential amino acid is an amino acid that has to be in your diet. Yes?
One of the other figures had histidine in that group.
Mm-hmm.
Is there...
A lot of these, and it's a good question. So we could actually, so if you put histidine in here, you say, well, how come, for example, aspartic acid isn't in this group? Because I say I could take an amine off of here and make aspartic acid. So it really depends. There's sort of artificial layers that are here. In a way, we could link this with almost any amino acid, depending on how much of it that we use. So the groupings that we see have to do mostly with how much of the amino acid is involved. Okay, so it's a good question. But amino acids are very interlinked, and it's difficult to say, well, these are the only ones that are involved in glutamate. Make sense?
Yeah.
Okay. Now, here's aspartic acid. There's oxaloacetate. That is an alpha-keto acid. Put an amine group on it. We've got aspartic acid. Aspartic acid can go to asparagine. Asparagine is just putting that amine on that R group carboxyl, just like we saw for glutamine. Aspartate can go to methionine. Aspartate can go to threonine. Threonine can go to other things. And aspartate can go to lysine. I knew that was coming. Do you want me to know all these? If I say no, you're going to quit listening to me.
Do you want to learn them as you're presenting them now, as opposed to other resources that may have them grouped differently?
Let's see how we get through it. We'll decide. Okay? There's always a video. If you want to sit and watch, you can watch. And then if I say, yeah, you got to do it, you come back and you watch the video, right? Nothing says you have to write this down right now. All right. 3-phosphoglycerate. Where do we see 3-phosphoglycerate? We've seen it several places. Where have we seen it? Glycolysis, gluconeogenesis, and? Saw it yesterday. Calvin cycle? Right? Okay. You actually also saw it in pentose phosphate pathway. Okay. 3-phosphoglycerate's floating around the cell. There's a lot of it available. 3-phosphoglycerate can very readily be made into serine, and serine can be made into these other two amino acids here. Aromatics, okay? Now, the aromatics are the most complicated of all of them in terms of synthetic pathways. If I were to show you all the steps involved in doing these guys, you would probably walk out on me, okay? There's a lot of reactions involved in making some of these amino acids. But they all have common roots, and the roots are interesting. The roots are phosphoenol pyruvate, which we see, of course, in glycolysis gluconeogenesis, and erythrose 4-phosphate. Where have we seen erythrose 4-phosphate? Oh, man. That's almost a trick question because I only briefly mentioned it, all right? You see it in, what does erythrose sound like? We can reason this out. It's what? So ose on the end means what? Sugar, okay? And sugar metabolism, what have we seen? We've seen glycolysis. We didn't see it in there, right? We saw gluconeogenesis. We didn't see it in there. Glycogen metabolism. We didn't see it in there. What's the other sugar pathways we talked about? Glycogen synthesis, not in there. There's another pathway that has ose in it. Pentose phosphate pathway. Okay, so remember when I talked about the pentose phosphate pathway, I told you the pentose phosphate pathway was a way of mixing and matching sugars. You can take 4, 6, 7, 3, back and forth, and this allows the cell to balance and make things that it needs. And if the cell is needing these amino acids, look what it's going to do. It's going to balance it in terms of making erythrose 4-phosphate so that that can be used to make these amino acids. Okay? All right. The histidine family. Okay? Histidine comes from ribose 5-phosphate primarily. Histidine is very complicated also, and we can see pieces of it coming from a variety of places. Where do we get ribose 5-phosphate from? Pentose phosphate pathway. Okay. All right. Now, in answer to the earlier question, I am not going to make you tell me which family gives which amino acids. Okay? That's just basically something for you to memorize, and I don't think it's really useful for you to do. All right? I think you should know the families. I think the families, the names of the families, are important because they are the sort of headlines through the sort of funnels through which the other ones are made. So I think knowing the families is useful. The glutamate family, the aspartate family, serine, pyruvate, aromatic, and histidine. Okay, and I'm not sure I put pyruvate up there, but there's pyruvate right there. Sorry, pyruvate right there. Valine, alanine, leucine. Okay. All right. Now, interestingly, there's a fair amount of interaction of the amino acid metabolism with the citric acid cycle. This is true both in the synthesis of amino acids and also in the degradation of amino acids. You may remember when I talked about both glycolysis and the citric acid cycle, I said that they were central metabolic pathways, meaning they're central to many processes in the cell. And we probably see no better example of this, the central nature of the citric acid cycle than we do with respect to amino acid metabolism. Now, again, I'm not going to ask you to reproduce this figure, so I don't want you to worry about that. But there's some things here that I think are really important to see. Some of the basic things that I've talked about with respect to transamination, I think you should know. I think you should know pyruvate and alanine, transamination. I think you should know aspartic acid and oxaloacetate, transamination. I think you should know alpha-ketoglutarate and glutamic acid, transamination. Very simple ones that I've talked about. You should also know that glutamine comes from glutamate by adding an amine. And you should know asparagine comes from aspartic acid by the same process, adding an amine. Now, the reason I mention these is they're largely what you see on the screen. There are many other things as well, but they're more complicated. All right, here's glutamine, glutamic acid. Okay, these guys coming into, this is the breakdown on the left, catabolic reactions. These guys break down ultimately into alpha-ketoglutarate. Well, it's no surprise for these two because they're just basically losing some amines and bang, they are alpha-ketoglutarate. All right. If we look over here at pyruvate, we see that these guys are broken down into pyruvate. We see that pyruvate can be used to make these. Surprise, surprise, there's alanine right there. Okay? Acetyl-CoA involves many of these. These are broken down ultimately on the left. We see that these are breakdown. On the right, there's syntheses going on. These guys break down into this, and this guy gets made into these over here. Now, as far as the alpha-keto groups that I talked about, there's alpha-ketoglutarate, there's oxaloacetate, and there's pyruvate. The three easiest ones for you to know that involve alpha-keto interaction. Now, if you look up here, you'll see almost every amino acid, not every amino acid, but almost every amino acid is represented up here somewhere. Okay? It tells us that the citric acid cycle is both a way to burn things up when we need energy, so we can use amino acids to generate energy. And it also tells us that when the body has plenty of energy, it can take intermediates out of the citric acid cycle and use them to make amino acids for making protein. So the citric acid cycle is a very flexible pathway. It's what I referred to earlier in the term as anaplerotic. Anaplerotic means to fill up. Okay? It can be filled up by amino acids. It can also use intermediates to fill up other pathways, to make amino acids. So it's an anaplerotic pathway that's very, very important. Without it, we have no energy and we have no amino acids. Here's the transamination reactions I showed before. Okay? Here's glutamic acid. Here is its alpha amine. Here is an alpha-keto acid. This could be pyruvate, for example. I'm left with alpha-ketoglutarate over here. And now an alpha amino acid, in this case, it would be alanine if I started with pyruvate. It would be aspartic acid if I started with oxaloacetate. Okay? These reactions are catalyzed by general enzyme categories known as transaminases. And transaminases are interesting. They use pyridoxal phosphate as a coenzyme. That's a very important component. And that pyridoxal phosphate actually acts as a carrier of the oxygen and or the amine, depending upon which direction the reaction is going. I mentioned earlier that we could easily go from 3-phosphoglycerate to serine. You can see the steps involved in that process. And no, I'm not asking you to memorize these steps. I'm just showing you these for example. But basically, to go from here down to here, we have to do an oxidation. That's giving us the alpha-keto. Okay? We have a transamination that's going from here to here. And then from this transamination, we have to basically pop off the phosphate that's on there, and we've got serine. It's a very simple and easy set of steps that take us from a glycolysis intermediate to an amino acid. And that leads me to something else that's very important as we talk about and we think about amino acid metabolism, and that is that there are amino acids that are primarily metabolized in the citric acid cycle, and there are amino acids that are primarily metabolized in glycolysis. That's the two divisions that we give to amino acid metabolism. Okay? Amino acids that are, that are primarily metabolized through the citric acid cycle are called ketogenic. Ketogenic. Those that are metabolized through glycolysis / gluconeogenesis are called glucogenic. Okay? Glucogenic. Now, it's useful that we have both of those because we need glucose. And if we're eating a diet that really doesn't have enough glucose in it, but we get plenty of protein and amino acids, we can make glucose as we need. That's the sort of foundation of the Atkins diet. Okay? Now, I'm sure you probably want to memorize this cycle right here. Wouldn't that be a great one for... And no, you're not going to have a note card either, right? Everybody's smiling at that going, he wouldn't dare. Why do I show you this? This is a very important set of reactions. We're actually going to see a little bit of this later when I talk about nucleotide metabolism. Right? That there's a very important thing that cells have to be able to do, and that's to be able to transfer not only a nitrogen from one molecule to another, but cells also have to do what's called one-carbon metabolism, or single carbon metabolism. Basically, that involves transferring a single carbon from one place to another. We haven't really seen that so far. And there's a couple ways in which cells do it. One of the ways in which they do it is with an intermediate known as folic acid. Folic acid is a necessary nutrient. We supplement bread and cereals with folic acid. And one of the reasons that we do is in recent years it has become apparent that deficiencies of folic acid lead to deficiencies in neural tube development and severe birth defects. Folic acid is essential for those processes. Well, what folic acid is doing at the molecular level is it's allowing single carbon metabolism. Folic acid actually acts as a carbon transfer unit. So anything that involves single carbon metabolism, and there's a lot of things that involve single carbon metabolism, will be dependent upon folic acid. So you can imagine if you have a molecule that's really important for all kinds of reactions in the cell, and you don't have enough of that molecule, you're going to have some very severe consequences for that cell. If that cell's involved in the nervous system, yeah, you're going to see some birth defects, and that's exactly what you see with deficiencies of folic acid. Now, I'll say more about this folic acid scheme as I talk about nucleotide metabolism. Okay? Now, folic acid metabolism turns out to be important, or it turns out to be important. It clearly is important, but it turns out to be interesting when we think about design of antibiotics. Okay? Design of antibiotics. The reason that that's important is that bacteria make folic acid in a different way than we do. We get folic acid largely from our diet. We don't synthesize it from scratch. Bacteria synthesize it from scratch. So now you think that, well, if I can design a drug that messes with the synthesis of folic acid, I may preferentially harm organisms that need to synthesize it compared to organisms that already have it in their diet, like we do. Okay? and that is the basis of what are called sulfa drugs. Okay? Sulfa drugs, as noted here, have a generic structure something like this. There's para-aminobenzoic acid. Anybody know where you get this from? Anybody ever hear of sunscreen? PABA free? That's what PABA is. Yeah. Okay. Now, PABA, all right, has some nasty effects on some bacteria because it can interfere with their ability to make folate. All right? It can interfere with the ability of the bacterium to make folate and can be used for that purpose. It doesn't affect us because, again, we are not making folate. We're getting it in our diet. All right, here's an example of a one-carbon reaction that's happening involving a folate. The difference between serine and glycine is this guy right here. Glycine, you recall, is the smallest amino acid. Serine is pretty close to it, the difference being this guy right here. In single carbon metabolism, a carbon, and that carbon can be, in this case, a CH2OH. In some cases, it can be a methyl. In some cases, it can be a ketone. It varies depending upon what's going on. That single carbon can be transferred to a folate and create a folate now that has an extra carbon. Okay, so we see what's happened here is we have something that isn't quite yet a ring. It's got 1, 2, 3, 4. But when this guy grabs a carbon, that carbon makes up the 5th member of the ring. Okay? This guy can now go take this carbon and donate it somewhere else. Kind of like we saw with the with the transaminase reaction, where it's picking up a nitrogen, donating it somewhere else. This guy's picking up a carbon, and it's going to go donate it somewhere else. We'll see one of the somewhere else's in nucleotide metabolism. Cysteine turns out to be very readily synthesized from serine. Again, I'm not going to ask you this, so just relax. It's kind of a relaxed day today. Serine is readily made from cysteine is readily made from serine. Cysteine, of course, is the only amino acid that has a sulfhydryl group. And if we look at this, we see the difference is right here. Here's an OH. Here's an SH. Okay? And serine gets that S from an S minus that it gets in this reaction, as you can see right here. It's kind of a complicated process of getting to here, but going from here to here to here is actually pretty straightforward. Just like we are dependent upon microbes for nitrogen, in many cases we are dependent upon microbes for the assimilation of sulfur from the environment, and some of those processes that happen in plants and bacteria that you can see happening right here happen to provide this sulfide. That should be an S minus 2. There's a minus sign missing right there. Okay. Now, another carbon, this is an important one, another molecule that's very important in single carbon metabolism is called S-adenosyl methionine, or SAM. Right? S-adenosyl methionine also participates in single carbon metabolism. It almost exclusively, in fact, it does exclusively handle only methyl groups. There's the methyl carbon right there. All right? Here's what methionine looks like. If I take that methionine and I attach it to an adenine, okay, what I have is S-adenosyl, that's the adenine part of it, methionine. And that little carbon right there really doesn't want to be there. The reason? Because this, as it's drawn, actually has a positive charge. This is a very good leaving group. It wants to get donated somewhere else. And S-adenosyl methionine will donate that methyl to a variety of reactions. You've actually seen it once before in the class when we talked about the synthesis of choline. Choline is made by 3 methyl groups that come from S-adenosyl methionine, and this is the source of those guys. Okay. Going through this fairly quickly. Essential amino acids, I mentioned them earlier. Okay. Now, tables that you see for essential amino acids will vary sometimes by 1 or 2 amino acids, and it's partly because some of these we can synthesize in limited amounts. All right. And so the question is, well, do we synthesize enough to make it non-essential or not? And that's why you see some variations between them. But in general, there's about 10 amino acids that we consider essential and about 10 amino acids that we do not consider essential, meaning that if they're not essential, we can make them. If they're essential, we have to have them in our diet. All right? Now, I'm not going to ask you to memorize these, but I just want to point out a couple things in them. Look over here in the non-essentials. Okay? The non-essentials include things that are readily involved in transamination reactions. Alanine from pyruvate, asparagine from aspartic acid, aspartate from oxaloacetate, glutamate from alpha-ketoglutarate, glutamine from glutamate itself. Okay? Those guys right there are simple transamination reactions. They're non-essential amino acids. We can readily make them in our diet. If we look over here, here's some of the amino acids I said had fairly complicated synthetic pathways, and those pathways, in many cases, don't even exist in us. So we have to have these in our diet. There's the aromatic phenylalanine. Okay? There's tryptophan. All right? It turns out that tyrosine, which is, where's tyrosine? Right here. Tyrosine is frequently listed as non-essential because we can make tyrosine from phenylalanine. So we kind of cheat. All right? Tyrosine is, by extension, not an essential amino acid because its precursor is an essential amino acid. All right? If we don't have this guy, we don't have this guy either. Okay? All right. So, and again, if you look from organism to organism, you'll see some differences also. This is in human beings right here. Okay. Here's what I referred to before as the glucogenic and ketogenic amino acids. Again, not asking you to memorize this. I'm just presenting this information for you. I think you should know what glucogenic means. I think you should know what ketogenic means. All right? Glucogenic amino acids, there's a lot more of them that are only glucogenic. Okay? There's only 2 that are only ketogenic, and there's a handful of them that are both. Okay? All right. Now, I've been talking about amino acids, and I have to stop and think about, well, I have seen now that amine groups can move from one amino acid to another, but I also saw that ammonium ion could latch onto something. Right? If I've got ammonium ion, I can grab a hold of it. Well, does that mean that I can also release free ammonium ion? And the answer is, yes, I can. All right? Yes, I can. And ammonia, or ammonium ion, is toxic. So we have to be careful how much ammonium ion that we have in our bodies. Our body does a pretty good job of doing that, but our body has to balance ammonia metabolism. So what I'm getting ready to tell you is the way in which body balances ammonia metabolism. Our body has to really maintain a balance. If we're getting too much ammonia in our diet, we need to excrete more. If we're not getting enough, then we have to be holding on to what we've got. Well, the balancing agent in our body for ammonia is urea. It's what we excrete in our urine. Okay? And the amount of urea that we excrete is partly a function of what's our balance of nitrogen in our diet. Another compound involved in this is uric acid. Okay? Now I'm going to show you the urea cycle in just a minute. But uric acid is a very interesting and important compound with respect to nitrogen metabolism. All right? We'll see that uric acid is involved later in the breakdown of nucleotides. It's involved in the breakdown of nucleotides. And if we look at this guy, we see, well, there's a nitrogen, there's a nitrogen, there's a nitrogen, there's a nitrogen. That's a very cool way of carrying a lot of nitrogen. All right? It is. Some organisms actually use uric acid as their excretory nitrogen molecule instead of urea. Birds, for example. Birds excrete uric acid instead of urea. And as a consequence, birds don't really urinate. They make a sort of a gooey mess. All right? And that gooey mess is partly a function of the fact that uric acid is not very water soluble. Okay? It's not very water soluble. And that turns out to have very important implications for human health. Because our bodies are making uric acid as well. We make it as part of nucleotide metabolism breakdown. All right? Well, what's that have to do with human health? All right? I said that uric acid is not very water soluble, and that's the clue to one of the problems with uric acid with human health. We don't excrete uric acid. And by the way, an odd organism that excretes uric acid, Dalmatian dogs. All the pre-vets in here may know that. Dalmatian dogs excrete uric acid. They don't excrete urea. And Dalmatian dogs tend to have a lot more health problems in terms of pain. Anybody ever have a Dalmatian? A lot of pain? Cranky?
Yeah, they go deaf, right?
Yeah. They have a lot of problems because they're basically handling a lot of uric acid. Well, when we have too much uric acid in our body, we create a real high potential for pain. You develop a condition known as gout. Okay? And gout happens because of the fact that uric acid is not very water soluble, and it will crystallize. And where it will crystallize is usually in the lowest point in your body. So the first signs that you have gout arise with an enormous pain in your big toe. Enormous pain. You see, in the old movies, they used to make fun of this. That was hilarious. You know, they grab their toe and so on and so forth. What could be grabbing their toe about? Well, that's what they're grabbing their toe about, is the fact that this crystal of uric acid is crystallized in a nerve cell, and that ain't good. Okay? So that's a real problem. Okay? Gout is fairly treatable today. There are drugs that will reduce the synthesis of uric acid and alleviate a lot of the problems with it. But there's another very interesting side to the gout story in human beings, and it's not understood how or why. All right? The observation is that people who have gout tend to have a lower incidence of multiple sclerosis. As far as I know, nobody has a really good handle on why that's the case, but it's a known observation that that is. The speculation that I have read about this is that uric acid tends to act like an antioxidant, and perhaps there's an antioxidant property of uric acid that is preventing some of the symptoms that happen with respect to multiple sclerosis. Okay? So, uric acid, a very interesting compound. Well, we don't, for the most part, have too much problem with this. We do most of our handling of nitrogen through urea, and there's a cycle that we will see that is involved in that process. Okay. There's uric acid. If everything is working okay, uric acid goes down to here, down to here, down to here, and ultimately we get urea. So uric acid can be a precursor for urea, and that's why it appears in these metabolic pathways. If we were to start up here, what gave uric acid, we would see that the breakdown of purine nucleotides, adenine and guanine, will give rise to uric acid. All right. Now, the last thing I'm going to say today in terms of biochemistry has to do with the urea cycle. Okay? The urea cycle is, like the citric acid cycle, circular in nature. Okay? It occurs in two places, as you can see. It occurs in the cytoplasm, most of it, and part of it occurs in the mitochondria. Okay? Now, as we go through this cycle, I'm not so much hung up on, certainly not on the structures of the molecules, and for the most part, I'm not hung up on most or many of the molecules, but there are some of the molecules I think you should know in this cycle. I'm not going to ask you what goes to what goes to what, but I think you should know certain things that are in the urea cycle. Okay? Urea is an important one, obviously. That's kind of a gimme. What's one of the things, if I say on an exam, what's one thing that's in the urea cycle, you can have a free question, right? So anybody that doesn't get that one, you know, I frequently, and I think I did this on you guys, I frequently have a question on my first exam that says, name all the amino acids that don't have nitrogen. And I put that on there as a gimme because amino means nitrogen, but many people start listing amino acids that don't contain nitrogen. Anyway, all right, so, but the urea cycle does contain urea. All right. Now, what's happening here? Okay. Well, the urea cycle is really here to balance nitrogen. It's here to balance. So we think of it as only going in a cycle, but in fact, things can come in and drop something off and then leave. All right? So here's something coming in and dropping something off. Aspartate is dropping in. It's participating in the cycle. When it drops in, AMP drops out. And then later, aspartate loses its amine group and comes out ultimately as fumarate. Okay? So we see coming in, going out. And we see intimate relations again with things associated, in this case, with the citric acid cycle here. We see it involved with nucleotide metabolism over here. And we see it splitting out urea so that we can get rid of this excess nitrogen that we have. Look at urea. It's got 1 carbon and it's got 2 amine groups. It's a good way of carrying nitrogen out of the body that you don't want to have there. Urea is very water soluble. That's why our urine is liquid, not sort of semi-solid like a bird's excrement. Okay? And it goes out. All right. What else do we have here? We have ornithine. Ornithine is a non-protein amino acid. Look at it. Carboxyl. There's an alpha amine right there. Ornithine doesn't appear in proteins. Okay? Ornithine moves into the mitochondrion, and inside the mitochondrion it gets converted into citrulline, and citrulline is also a non-protein amino acid. Okay? Yeah. Ornithine is O-R-N-I-T-H-I-N-E. Citrulline is C-I-T-R-U-L-L-I-N-E. Okay? Notice the involvement of arginine. Arginine, and I think this is important, is the immediate precursor of urea. All we have to do is cut this guy, cut arginine right there on one end, and when we cut it, what comes off is urea, and what we're left with is ornithine. So basically, arginine minus urea equals ornithine. Okay. Now, what else do I want to say here? I want to do that kind of quickly. I think that's mainly it. This is very important for us to be able to have a way to dump out this urea. So if we have a lot of ammonia, or we have a lot of nitrogen in our body, more than we need, this guy is going like crazy, and it's splitting off this all the time. If we don't have as much of that, this cycle is not going to be going on very much, and we're not going to split off very much urea at all. Okay. I thought we would finish the day with a song. What's that? Somebody say extra credit is what he said? Yeah.
Everybody comes to the second last day. Everybody comes to the last day. No one comes to the second.
That looks like good attendance to me. So this is actually a video that I put on YouTube for one of my songs a couple years ago. I thought I'd play it here. If you want to hear it, great. If not, that's fine. I'm not hurt if you don't listen. It's called Around the Nucleus. Oh, come on. That's better. I won't say what I was on when I wrote this one. Okay. non-protein amino acids.
Where did, I thought there was 20.
There are 20 that appear in proteins,
Oh, okay.
but there are non-protein amino acids.
Okay.
So it means that they're not.
Yeah, I thought
There are
there
20
was
that appear in
20.
proteins. That's right.
Gotcha.
But there's a difference, right?